I haven't scheduled a review session yet. I will let you know on Friday when I schedule that. I'm thinking probably about Monday evening would be my guess. Yes? On Monday evening? How many have Monday evening exam? What time is the exam done? Oh, we can start doing it at 10. What do you guys think? That was a joke. That was a joke. Okay, so the exam is on Wednesday. That would probably mean I would need to do it on Sunday. Let me think about that. I can't do it Saturday, so it would have to be Sunday. Well, the problem if I want to do it in the morning is that there's always going to be exam conflicts with that. So I try to minimize conflicts as much as I can. So I will shoot for Sunday evening. Probably early evening would be my gut feeling. I'm thinking about 5 o'clock or something like that. But let me look at my calendar and look at the availability of classroom space and get that set up. I'll get that set up before next time, so I'll let you guys know when that is. Okay? Yes. The exam will be right in here. Yep.
Did you say something last week regarding note card?
I'll be handing out a note card that you can use. You have to get the note card from me, and you have to turn in a note card that you got from me even if you don't plan to use the note card. So that's a requirement for the final exam. I will bring the note cards next time. So, yes, sir.
Will your office availability be different?
Yeah, I'll probably be there more, and I have fewer conflicts. So during finals week, those of you who have seen my schedule know it's pretty crazy. During finals week, I don't hardly have anything that's conflicting. So I'll be available. I'm more than happy to meet with any or all of you that want to come by. I have enjoyed working with many of you on, well, not many of you, all of you who've come in on the exam, and that's been very good and informative for me. So I appreciate you guys coming in for that. You'll get the exams back on Friday. Okay, so the exams will come back to you on Friday, and I'm working on those right now. So it takes quite a while to get back through all that, but you'll have them on Friday. Okay, well, what we had to talk about here that we haven't talked about in photosynthesis are the dark reactions. So the dark reactions are the second phase of photosynthesis. You've seen how the excitation of electrons by photons of light lead to the transport of protons into the thylakoid space, and it's that gradient of protons that leads to the synthesis of ATP. And you've also seen how the ultimate electron acceptor is NADP to create NADPH. And so that's a very important consideration, particularly as we get to the Calvin cycle. So let's take a look at the Calvin cycle. Now, you're just going to love this cycle. No, you're not either. Okay. And we're not going to do it in much depth. So I focus on a couple things in the Calvin cycle. If you take a majors level biochemistry course, everybody gets excited and pees their pants over how many of this goes to how many of this goes to how many of this. And it's really just bean counting. Okay. It doesn't really matter. If you look at the reactions in here, and I'm not going to go through the reactions in here, and you're not responsible for the reactions in here, except for just a couple that I'll talk about, you'll see that many of these are reactions that you've seen before. Some of the enzymes that we see here are similar, or are the same enzymes that we see in the pentose phosphate pathway. So you'll see transketolase, and you'll see transaldolase in the Calvin cycle, because these enzymes are involved in moving, my head feels like there's something on it. These enzymes are involved in moving one or two pieces of carbon from one molecule to another, as they were in the pentose phosphate pathway. So there are some similarities between the Calvin cycle and the pentose phosphate pathway. Now, the bottom line of what's happening in the Calvin cycle is that carbon dioxide is being taken from the atmosphere, and it's being built into a molecule. Now, when I talk about it being made into glucose, that's a little misleading, because there's no sequential this carbon plus this carbon plus this carbon plus this carbon, and you get 6 of those together and make glucose. That's not how it happens. Instead, what we see is that some of the intermediate molecules gain a carbon, and then they get sort of rearranged. So through the mass action of all of that, a net glucose is produced. Right? And if you don't understand that, don't sweat that too much. The bottom line about what is happening in the Calvin cycle is carbon dioxide is being taken from the atmosphere. So that's where I'm going to focus things at first. Okay? So what happens in this process? What happens in the process of the Calvin cycle is that we have a starting compound called ribulose 1,5-bisphosphate. And you can call that RU15BP. Ribulose 1,5-bisphosphate is a 5-carbon sugar. So it has a phosphate on the first, and it has a phosphate on the last. Okay? Ribulose 1,5-bisphosphate. Okay? The very first reaction of the Calvin cycle incorporates that carbon dioxide. And you can see that carbon dioxide coming in right here. So if I start with a 5-carbon piece and I add another carbon to it, I'm going to make something that has 6 carbons. And it turns out that this 6-carbon piece is very unstable, and it immediately breaks down into 2 pieces of 3-carbon. Okay? The 2 things it breaks down into are 3-phosphoglycerate. And yes, that sounds like something you've heard before, because 3-phosphoglycerate was an intermediate in glycolysis. Now, I'm not going to go through the subsequent reactions of that because that just confuses the picture. Right? I'm just going to point them out in general ways to you in a minute. But you should know the enzyme that catalyzes this reaction. This is what has been described as the most abundant enzyme on the face of the earth. I'm going to give you the full name, then I'm going to give you an easy-to-remember shortening of it. Okay? The full name is ribulose 1,5-bisphosphate carboxylase. Ribulose 1,5-bisphosphate carboxylase. The shortened name that most people call this enzyme is called RuBisco, R-U-B-I-S-C-O, and you're welcome to call it that. RuBisco. Now, RuBisco is very likely the most abundant enzyme on the face of the earth because plant cells are full of it, because they have to incorporate carbon dioxide in order to make glucose. And plants, as you know, are everywhere. Okay? So this enzyme is very abundant. Now, this enzyme is an unusual enzyme, and I'll say a little bit more about it later, in that it's very inefficient. It's very inefficient. And we'll see how its inefficiency leads to some unusual circumstances for plants. All right, so RuBisco, very important. RuBisco is the one that incorporates the carbon dioxide. Now, if you look at the rest of this picture and you follow the arrows and you see all the interplays and you look at all the numbers, okay, all this is showing is that if we start with, let's say, 6 of these, we've incorporated 6 molecules of carbon dioxide. And through a lot of rearrangements, which is what's happening through all of this, a net molecule of glucose is produced. That's all that's there. So you don't have to worry about how that net molecule of glucose is produced. You don't have to worry about all those rearrangements. But if you look carefully on there, you will see that there are intermediates from glycolysis, and there are intermediates from the Pentose Phosphate Pathway, and there are enzymes that are common to both. And we see things like gluconeogenesis going on in here. All of these things contribute to making that molecule of glucose. And that's all there is to it. Okay? So the incorporation of carbon dioxide by RuBisco leads to the production of glucose within plant cells. Okay? Here's the actual reaction. Okay? There's the RU15BP. There's that 6-carbon intermediate. You don't need to worry about that, but the 6-carbon intermediate that is there, it's unstable, and it falls apart into 2 molecules of 3-phosphoglycerate. So we start with 5 carbons. We now have 6 carbons. And if we have 6 of these, we have 12 of these. And we have, therefore, a net gain of 6 carbons. And that's what it takes to make a glucose. Okay. This is the structure of the enzyme. To be honest, I'm not sure why I put that up there, but it's a nice-looking enzyme, right? And this is a bit more information about the enzyme. It is, as I said, very inefficient at what it does. So let's take a brief look at this enzyme. Yes, okay. This reaction scheme shows something that surprised people when they first discovered it. And it's still surprising, and it's still a bit of a puzzle. The reaction scheme I showed you before showed you that RuBisco can take carbon dioxide and add it to Ru-15BP and make 2 molecules of 3-phosphoglycerate. However, it turns out that RuBisco can also be confused by molecular oxygen. Okay? Molecular oxygen. And this molecular oxygen is what makes this enzyme very inefficient. And it's not known anywhere that any of the RuBisco enzymes in any plant have evolved a way around this. Well, why is this important? Well, instead of incorporating carbon dioxide, it's incorporating oxygen. And when you incorporate oxygen, you're not going to get an extra carbon coming in. You're going to get oxygen coming in. And when that guy falls apart, you don't get 2 molecules of 3-phosphoglycerate. You get a phosphoglycerate and a 3-phosphoglycerate. Notice this guy has 2 carbons. This guy has 3 carbons. Okay? Well, why is that important? Well, it turns out that this guy falls apart, loses the phosphate, and becomes, look at this, glyoxylate. Where did you see glyoxylate before? What'd you say? The glyoxylate cycle, right? And where did we find the glyoxylate cycle? What's that? What organisms? Plants and bacteria and yeast, right? Okay. We don't find this in animals. Well, it turns out glyoxylate is a pretty poisonous compound. It's not good for cells. Here's an enzyme that is making a compound. This, of course, is useful, but it's making a compound that is detrimental to the cells. It makes sense that you have a cycle that can gobble up glyoxylate. So the glyoxylate cycle in plants makes very good sense. Now, it's inefficient because we're seeing that we're starting with phosphates and we're actually losing a phosphate over here. And you know putting phosphates on frequently requires ATP. So going through this cycle here is basically wasting energy and not making anything very useful. Okay? So that's part of the reason why this enzyme is described as inefficient. It doesn't it does preferentially take carbon dioxide, but it will take this with pretty reasonable frequency. Okay? Yes, ma'am.
Does the oxygen get incorporated every time? Or
Well, if the enzyme binds the oxygen, yes. So it's a question of what the enzyme binds.
So like if the enzyme doesn't bind O2, then it's not an inefficient enzyme? Or
Well, let's imagine that the enzyme, if it only binds carbon dioxide, every time it binds something, it's going to incorporate a carbon. But if 50% of the time it incorporates carbon dioxide and 50% of the time it incorporates oxygen, then 50% of the time it's going to be wasting energy, right? So it depends on how frequently it binds that, but it means that it's not going to be not going to be as efficient as it would be if it only bound carbon dioxide, right? Good question. So this is unfortunate. There are a lot of people right now interested in genetically engineering RuBisco, this guy here, to make it more efficient and not do this reaction. Okay. Now, The last thing I want to talk about is something that some of you no doubt know a fair amount about, and that's what's called C4 plants. So in order to tell you about C4 plants, I need to first of all tell you about C3 plants. So the reaction I showed you for the Calvin cycle is called a C3 reaction because the primary product that comes out of that are 2 molecules of 3-phosphoglycerate. Okay? Now, I'm not a botanist. Some of you in here are botanists, so you'll be better in position to talk about which plants are C4 compared to C3. But C4 plants have a fundamentally different mechanism of doing the dark cycle than C3 plants do. They still do the Calvin cycle, but they have a cool trick that they have evolved in order to avoid the loss of water. Okay? So C4 plants are pretty efficient at not losing water. And the reason that they have that's the case is that they do the incorporation reaction not in the outer part of the plant, but instead internal to it. Okay? So it's internal to it. So the reaction of the Calvin cycle are occurring in here away from the outer part of the plant. To get this guy into here, okay, and if I were to do these reactions out here, all right, I would be exposing a lot more of the aqueous environment of this cell out here to the atmosphere, and it would evaporate. So these plants have evolved an ability to bring in carbon dioxide without exposing much water, so they don't lose much water by evaporation. But the problem with that is that the carbon dioxide comes in here, but the Calvin cycle is occurring way over here. Okay? So what the C4 plants have evolved is a way to incorporate that carbon dioxide temporarily into oxaloacetate. There's our friend again. And oxaloacetate, you remember, has 4 carbons. This is why we call this a C4, because the very first product that's made when carbon dioxide is bound is made into a 4-carbon compound, oxaloacetate. Now, all that oxaloacetate is doing is holding on to that carbon dioxide so that it can be transported further into the plant and then be dropped off into the Calvin cycle. Okay? So it's a delivery mechanism. Now, this feature is common to all C4 plants, and all C4 plants are therefore better able to manage water. And they're very good at incorporating the carbon dioxide, because if you think about it, we're sort of concentrating all of that in here from something that's very diffuse out here. So C4 plants are very, very efficient plants. Yes, ma'am?
So I guess, so the Kelvin cycle's the dark phase, right?
Yes.
And then, so I guess, when we went over that, I didn't really catch where the water is lost.
So the water is simply lost by the openings in the cell that's necessary to grab this stuff out. Okay? So this has evolved, these plants have evolved a way to reduce those openings, get the carbon dioxide, and then do the Calvin cycle away from where the outer part of the cell is. Yes, sir? They're commonly found in tropical environments, and they're also found in fairly dry environments as well. Yes? Now, let's follow what happens here. Carbon dioxide comes in. There's our friend PEP. Okay? PEP was made into oxaloacetate in which pathway we've talked about? Gluconeogenesis. Point of extra credit. Okay? Gluconeogenesis is where we saw that happen, right? That was the pyruvate carboxyl. I'm sorry, that was the PEP. Actually, I take it back. We actually did the reverse reaction, oxaloacetate, to that using what enzyme? What was it? PEPCK. That's right. Point of extra credit there. All right? So we did the reverse reaction. We converted oxaloacetate into PEP, so then we could go all the way up the cycle in the gluconeogenesis. Right? All right. This guy's making oxaloacetate. all right? And it is then reducing oxaloacetate to malate. So we're doing two backwards reactions here. Backwards reaction of gluconeogenesis. We're doing a backwards reaction of the citric acid cycle. And we do that because malate can be transported into here. Notice I'm not giving you enzyme names. Okay? Malate being transported in can lose its carbon dioxide, and this is the delivery mechanism to put the carbon dioxide into the Calvin cycle. So in here is where RuBisco is going to be. Then we see the Calvin cycle. Okay. Pyruvate's released, gets transported back out here, and we go through the whole cycle again. Yes, ma'am? Malate has 4 carbons. You have 3 carbons, 4 carbons, 4 carbons. Then it goes over here, 4 carbons, loses 1, becomes 3 carbons again. Okay? Good. Okay. And there's all the reactions. You don't need to know the enzymes. Okay? I'm not going to hold you responsible for the enzymes. Okay. So we have finished photosynthesis. We are now moving on to our last topic. Hallelujah, right? So nitrogen metabolism is an important and interesting topic. Okay? Nitrogen, of course, is necessary for amino acids, and amino acids are necessary for protein. Nitrogen is also necessary for things like nucleotides, and we'll talk about nucleotide metabolism here. But if we look at nitrogen in the environment, we see something that looks like this. Okay? We have atmospheric nitrogen, which is N2, which is the most abundant gas which is in our atmosphere, and we can't do anything with it. Okay? We can't do what's called fix nitrogen, meaning we can't incorporate nitrogen into molecules. We are completely unable to do that. And in fact, most organisms on Earth cannot do that, which means that the nitrogen that we have that's being used for amino acids and so forth is being incorporated from the atmosphere by a very tiny number of organisms. I want to emphasize that. A very tiny number of organisms. And they're almost all microbial. Okay? They're called nitrifying bacteria. Okay? Nitrifying bacteria. We see them commonly associated with the roots of some plants, like legumes. We see some of them as free-living bacteria. Okay? But I tell you this because every time I tell people this, it makes me worry. All of the nitrogen on Earth that's found in biological organisms is needed. We need these tiny organisms to do it. And when we think about water pollution and air pollution and soil pollution, we think about endangering those organisms. We think about endangered species. We don't talk about these guys right here. But as we endanger them in any way, we are endangering the nitrogen of the entire planet. That's pretty serious. Right? That's pretty darn serious. And we don't talk about it. I talk about it. I scream and holler about it. Because I think we should all be aware of this. Okay? We need this to happen. Now, there's a variety of things that they do. Some of them convert into ammonia. Some of them convert into nitrites, nitrates. We don't really care here for our purpose how that happens. But ultimately, we're wanting to reduce nitrogen to something like this that can be used to make amino acids. And we're going to focus, I'm not going to focus on these guys or these guys. I'm going to focus on this. Okay? And I don't care that you know how that's made. Okay? But I do want you to know that bacteria are critical for this process. Absolutely critical for this process. Okay. Well, how do they do it? They do it first of all, there's an enzyme that these bacteria contain that is a very unusual enzyme. It's called nitrogenase. Okay? Nitrogenase. Nitrogenase is able to take molecular nitrogen, add a bunch of electrons to it, add a bunch of protons to it, and make ammonium, NH4. Okay? This reaction is, first of all, completely impossible in the presence of oxygen. It has to be done in a completely anaerobic environment, which is why you frequently see these organisms in the in the roots of plants, because the plant roots provide an environment that lacks oxygen. That structure is necessary to exclude oxygen. The other thing I want you to notice here is look at the energy requirement here. To make this, we need 16 ATPs. Not we, the bacteria needs 16 ATPs. So we've got a very endangered kind of a thing. That oxygen kills it, and the organism has to invest a lot of energy into making ammonium that can then be used to make amino acids and nucleotides. So nitrogenase is a critical enzyme in that process. The overall process we call nitrogen fixation. And it's not that you have a fixation about it, but I guess you should have a fixation about it because it's a pretty important process. But nitrogen fixation involves taking atmospheric nitrogen and converting it into biologically useful nitrogen, like ammonium ions. Okay? Okay. Well, what I want to do now, I'm not going to show you the reactions about how that happens and so forth, because, as I said, that's kind of involved. But I do want to spend a little bit of time talking about the movement of nitrogen within and between organisms. We saw a little bit of the between that with the environmental picture on the last slide that or the two slides previous that I showed you. Nitrogen is both a precious commodity and it's a poison. It's a little bit like glucose in that sense. We need glucose, but too much glucose is poisonous to us. Okay? Nitrogen is the same way. Cells and our bodies have to be able to balance nitrogen. Okay? We have a cycle that we run called the urea cycle. And the urea cycle is critical for our nitrogen balance. We want to be able to use nitrogen as it comes in as we need it, and we want to be able to excrete it when the levels are too high. So all organisms have a means of balancing their nitrogen because of this very thing I just told you. They all have a means of balancing their nitrogen. Now, one of the simplifying things that we're going to see about nitrogen is that much of the nitrogen in our cells moves as a result of a very common reaction known as transamination. Transamination is a common way in which amines are moved from one molecule to another. Transamination. The enzymes that catalyze these transaminations are known as transaminases. And we're going to see they have a common mechanism that they use. But before I show you that, I want to start talking a little bit about the metabolism of amino acids, because this relates indirectly to start, as we will see, with nitrogen metabolism. The very first one I show you is going to be an enzyme called glutamine synthetase. And it's an enzyme that actually uses ammonium ion. Okay? It's not the amines that we see in amino acids, but it actually grabs a hold of ammonium ion. Now that turns out to be important because one of the ways in which cells deal with excess amines is to release ammonium. Ammonium is interchangeable with the word ammonia. Okay? You put ammonia in water, you get ammonium ion. You've all done freshman chemistry where you get, ammonium hydroxide, and whoa, that knocks you over, right? I mean, it's nasty stuff. That's just ammonia dissolved in water is all it is. Okay? Handling that ammonium is critical. Okay? Organisms are going to grab it and use it where they need it, and they're going to push it in the direction of making urea, if it's you and I, because that's what our bodies do, if they need to get rid of it. So all organisms, as I said, have a way of handling biological nitrogen. We excrete urea because it contains amines. And that allows us, when we have too much, to get rid of it. Other organisms have different things that they excrete. Okay? Birds, for example, excrete uric acid. That contains a fair amount of nitrogen within it. Fish excrete ammonia. That's why if you don't clean your fish tank out for a while, you get a really ripe odor of that. Okay? Because they're excreting ammonia, and you get that directly. All right. Well, let's get back to glutamine synthetase. Glutamine synthetase I start with because it's probably the central, most important enzyme in cells as respects ammonia and ammonium. Okay? It's a very complicated enzyme. We're going to focus on a very minor component of it. The minor component is that glutamine synthetase catalyzes the conversion of glutamate into glutamine. That is glutamic acid into glutamine. Okay? The difference between glutamic acid and glutamine is that the glutamic acid has a carboxyl group in its R group, and glutamine has a carboxamide group in its R group. Yeah. So glutamic acid has a carboxyl group in its R group, and glutamine has a carboxamide group in its R group. Now, we can see right here the ammonium ion being incorporated. There it is. If we're going to make a carboxamide, we've got to bring in a nitrogen. And here's the nitrogen coming in. This is one way for a cell to detoxify ammonia. Because if you have too much free ammonia sitting around, the cell dies. So one way to detoxify that is to put it onto glutamate and make glutamine. That reduces the ammonia concentration. This reaction, you can see, requires energy. And then here's the complicated part. Look at all of the allosteric inhibitors of this enzyme. Holy moly. You haven't seen one like that, okay? Many of these allosteric inhibitors are actually things that glutamine can be made into. Glutamine can go to all these things, and look how they feed back and inhibit this enzyme. The feedback inhibition is complex. One alone is not going to stop the enzyme. It's a combination that affects the enzyme. It's a combination that affects the enzyme. That's why we're not going to talk about the complications of that, because it's beyond the scope of this class. But suffice it to say that if all these guys are present, this enzyme is going to be inhibited. Okay? This enzyme is going to be inhibited if they're all present. That could be problematic, right? Because if this enzyme isn't working, what's going to happen with this? Its concentration is going to grow, right? Well, fortunately, our cells in our body have a way of shuttling some of these things out. Here's a good example. Alanine can be shuttled out, okay, and sent into the bloodstream where it goes to the kidney and dumps off its amine. And the kidney takes that amine, and what does it make with it? Urea. So you pee it out. Okay? So we have mechanisms of dealing with that. But as this thing starts to stop and this starts to accumulate, it's going to get sent out to the kidney. Yes, sir? The mechanism? It's just a toxic compound. If you have kidney failure, what's happening isn't the kidney that's doing it, it's the ammonia that's building up in all of your other cells. So that ammonia, the ammonia is reactive, so it can bind to a variety of things. One of the things that ammonium will do is ammonium will affect precipitation of proteins. Okay? So you could be doing all kinds of very drastic things inside of your cells. And so it's the accumulation of that as a result of kidney failure that actually kills you. Yeah. Ammonium can cross the blood-brain barrier. Yeah. Yep. Yes? Glutamine is not a feedback inhibitor. As you can see, it is a central molecule in many other pathways. Yep. Okay. So, complicated thing there. No, you don't need to know all these molecules and all of that. But I do want you to know that this enzyme is central to ammonium metabolism. I want you to know that it converts glutamate into glutamine. And I want you to know that it has a complicated regulation. Complicated regulation. Well, glutamate and glutamine, as you're probably starting to gather, play some really important roles in nitrogen metabolism in our body. Okay? I'm going to refresh your mind with structure here as to the differences between them. Alpha-ketoglutarate is intermediate in the citric acid cycle. And I told you when we studied the citric acid cycle that alpha-ketoglutarate can readily be made into glutamic acid. And that's what you see happening in this reaction right here. Okay? Here we have a double-bonded oxygen, and here we have a nitrogen. Okay? Yes, ma'am. Yes. Yes. Any acid is that way. So acetic acid, acetate, palmitic acid, palmitate, etc. People use the distinction, the ate on the end to refer to the ionic form and the acid form to refer to the proton on. I use them interchangeably. Glutamic acid, glutamate, same thing. Okay. Glutamate differs from glutamine in this group right here. Nope, let's see. Hold on. Right here, okay? So instead of having that COOH on there, we have an NH2. So this guy has gained an NH2 in the process. Now the difference between this and this is right there. Okay? So you can see that difference right there. Now this becomes important because we're going to see that particularly this reaction is involved in transamination. So let's talk about that. Let's see, do I have, oh blast it, I hope I didn't take that out of there. Yes, here's what I want to show you. Let's talk about, now that I've introduced glutamate to you, I've introduced glutamine to you. I told you that they're important in the movement of nitrogen. Now let's see how they're involved in the movement of nitrogen. This involves a phenomenon known as transamination, and you've seen a little bit of this already. Glutamate, or glutamic acid, shown right here on the right, has an amine that can be swapped with an oxygen of an alpha-keto acid. Well, what's an example of an alpha-keto acid? Okay, alpha-ketoglutarate was one. Oxaloacetate was another. Pyruvate was another. There's three right there. So let's assume for the moment this is pyruvate. Okay? If this is pyruvate, then that R group is a methyl group. Okay? Everybody with me? So we assume the R is a methyl group right here. In a transamination reaction, this guy and this guy are going to swap places. Look what happens. Glutamate gains an oxygen here, That makes alpha-ketoglutarate. This pyruvate gained an amine, and it has become alanine. Okay? So all that happened was the oxygen and the nitrogen swapped places on the two different molecules and created these two guys here. This transamination reaction happens across a wide spectrum of amino acids. They can start with an alpha-keto acid and become, okay, an amino acid. Here's glutamate with oxaloacetate as an example. There's the alpha-keto group right there. There's the amine. When they swap positions, I get alpha-ketoglutarate again, and I get aspartic acid. These reactions are reversible. If I have too much aspartic acid, I can take some of the alpha-ketoglutarate from the citric acid cycle, go backwards and make glutamate and oxaloacetate. So this transamination, the swapping of that nitrogen and that oxygen, allows for the movement of amines from one molecule to another. Very, very simple process. And as I said, these enzymes are called, you can see aminotransferase. I like to call them transaminase because it's very simple. Don't worry about the bigger name. Transaminase. That's all you need to worry about.
Thank you for sharing that.
Okay. Well, transamination, I hope I've planted in your head, is a really important process. Now what I want to do, and this is something that nutritionists oftentimes will be very interested in, or talk about families of amino acids. So one of the things you learn about amino acids in nutrition are that some amino acids are what we call essential, and some amino acids are what we call non-essential. I'm going to give you a couple definitions there. An essential amino acid is an amino acid that you have to have in your diet because you can't make it. Your body can't make an essential amino acid. Okay? In some cases, your body can't make it in sufficient quantities. Arginine is a good example of that. Your ability to make arginine varies between being an infant and being an adult. In some infants, arginine is actually essential, and in adults, it often is not. So when you look at the number of essential amino acids between adults and infants, you'll see that that number sometimes will vary a little bit for the reason I just stated. A non-essential amino acid, your body can make it. You don't have to have it in your diet. Okay? Your body can swap other things and make that okay. All right. Well, this guy here is called the glutamate family because these amino acids all can be made from glutamic acid. No, I'm not going to ask you which ones can be made from glutamate. I'm simply going to ask you to know the names of the families. Know the names of the families. So the glutamate family is right here. And we'll see that some of the families are named for the central molecule. Others are named for the starting molecule. Some people call this the alpha-ketoglutarate family, but I prefer to call it the glutamate family, okay? Because everything is made from glutamate. These are fairly, you've seen the reaction that goes to this. The reactions that go here and here are somewhat more complicated. Okay? But all of these things can be made from glutamate. What's the second family? Second family is the aspartate family. And you've already seen the relationship between oxaloacetate and aspartic acid because that's simply a transamination that gives rise to this thing. Aspartic acid differs from asparagine just like glutamic acid differs from glutamine. The R group here has a carboxyl, and the R group here has a carboxamide, just like glutamine did. Again, the synthesis of these guys is somewhat complicated compared to the synthesis of this guy right here. The third family, serine. That's misspelled, isn't it? Glycerate instead of glycerate. There's our friend 3-phosphoglycerate. Now you've seen it in several places. You saw it in glycolysis. You saw it in gluconeogenesis. You didn't know it, but you saw it in the pentose phosphate pathway. And now, today, you saw it in the Calvin cycle. Okay? And now you're seeing it's also a part of the synthesis of serine in amino acid metabolism. Serine leads to these two amino acids. And it turns out that the metabolism of serine and glycine is very closely linked to the synthesis of folates. Very closely linked to the synthesis of folates. And yes, I think you should know that. Folates are important dietary compounds. Okay? Folic acid is essential for development of neural tubes during embryogenesis. And I'll say a little bit more about that later. But that's the serine family. The pyruvate family. Again, pyruvate and alanine interlinked by transamination. Okay? Interlinked by transamination. So you should know all of the alpha-keto acids I've talked about. There are 3 of them so far. Alpha-ketoglutarate, which can go to glutamic acid. Oxaloacetate, which can go to aspartic acid. And pyruvate, which can go to alanine. Those are real easy transaminations. Yes, pyruvate, which goes to alanine. Yep. Yes, ma'am. So they're all alpha-keto acids. So alpha-ketoglutarate is one of the alpha-keto acids. So the alpha-keto acids are alpha-ketoglutarate, oxaloacetate, and pyruvate for our purposes. Okay? Okay, another family. As you can see here, there are 6 families. One family is a special case, as you will see. Here's the aromatic family. Okay? These all come from these 2 compounds here. PEP, there's our friend again, and erythrose 4-phosphate. A point of extra credit for whoever can tell me where they've seen erythrose 4-phosphate in a pathway. I'll give 2 points of extra credit if anybody knows. Because I don't think I've mentioned it, but you would have had to have noticed it. Okay. It's found in the pentose phosphate pathway. Okay. It's found in the pentose phosphate pathway. When I talked about the ability of the pentose phosphate pathway to mix and match numbers of sugars, 3 plus 4 goes to or let's say 4 plus 6 goes to 5 plus 5, for example. Okay. So the 4 is this guy right here. So, all right, that was a trick question. So the aromatic family there. The aromatic family, of course, includes phenylalanine, tyrosine, tryptophan. You know that from knowing your amino acids anyway, so I'm not going to say anything more about that. The last family is a special family because it only has one member, and that's histidine. And histidine looks like it's very simple. You go from ribose 5-phosphate to histidine. However, there are 10 steps in going from ribose 5-phosphate to histidine. It's not a fun pathway, let me tell you. Okay, so 6 families of amino acids that all are linked. And in each case here, we're talking about the synthesis of fatty acids. The synthesis, okay? The breakdown of fatty acids is commonly what yields ammonia. The breakdown of fatty acids commonly yields ammonia. So as we're breaking down fatty acids, we have to deal with that ammonia component. And this is something people don't talk about a lot either. Those of you who are interested in high ketone diets are burning amino acids to get glucose. You're burning amino acids to get glucose. What are you doing to your ammonium concentrations? Okay, keep that in mind. Some people are concerned about the effect of these on kidneys, for example, because high ammonium may be stressing kidneys too much to do stuff. All right, the last thing actually, I shouldn't. I'll talk about that next time. Let's save that for next time. I have a song about photosynthesis that I'm going to take an attempt to sing. I don't usually try to sing it, but you guys can help me. It's to the tune of an old song called Scarborough Fair by Simon and Garfunkel. It's also an old, I think, Scottish tune originally. So please join me. Photosynthesis is divine, fixing carbon using sunshine. It's thanks to plants that we've got a prayer. They pull CO2 from the air, reaping energy from the sun. It's efficient, second to none. You grab the photons almost at will. Protoporphyrin chlorophyll. Light reactions of system 2 split up water making O2. Electrons pass through schemes labeled Z, pumping protons gradiently. ATP's made due to a shift of the proton spinning quite swift. An enzyme turbine cellular maze, you know as ATP synthase. Carbons fixed onto a substrate, ribulose 1,5-bisphosphate. RuBisco acts inefficiently, splitting it into 3 PGs. If the enzyme makes NO2, it makes glycolate, it is true. The Calvin cycle works in a wheel, giving plants a sugary meal. So photosynthesis is divine, 'cause it happens all of the time, from dawn to dusk and times in between. Solar panels truly are green. Okay, guys, see you last class on Friday.